welcome friends, and again I come with my new topic, and after a just 2 days interval. And today's topic is molecular trafficking, or the transport mechanism of the components of nucleus, and why we know about the nuclear transport and what is the importance of the nuclear transports. Then firstly, it must be known that the nucleus as the brain of the cells, thus the transport mechanisms of the nucleus is very important, because the molecules which are exported or imported from or outside or inside the nucleus, this is very important for the other cellular metabolism and other cellular regulation and fine tuning of cell signaling, and etc etc. So thus the nuclear transport mechanism is a very important topic, and in today's molecular biology, for detailed knowledge and for higher studies of molecular biology, let's get started. So friends, you are clearly seen that these are the diagrams of the nuclear transport mechanisms, and there are several numbers, means the 1, 2 and 3 and the 4. The 1 number denotes the overall mechanisms of the nuclear transports, means the how the GTP hydrolyzes the GDP, and what is the concentration of the GTP and what is the concentration of the GDP in both the outside as well as the inside of the nucleus. And the number 2 picture denotes that the import mechanisms of the nucleus. Yes, this is the import. And the number 3 pictures shows that the export mechanism of the nucleus. And the number 4, it is the mechanism of transport of several transcription factors like NFKB and phosphor. Phosphor is the transcription factor of the yeast, etc etc. And then let's get started the explanation of these diagrams. Firstly, the number 1 pictures show that the GTP, which is bound with the several proteins, and the GTP is hydrolyzed into GDP, and in this hydrolysis Ran-GEF means the guanine activating protein. Guanine is activating protein and is help in this kinds of transformation, means the GTP to GDP, and thus the GDP concentration is higher inside the cytoplasm, and obviously the outer side of the nucleus, means the concentration of the GDP is higher in cytosol rather than in the nucleoplasm. But the reverse case is occurred in case of inside the nucleoplasm, and in this case the GDP is hydrolyzed, or sorry, GDP is transformed into GTP, and thus the concentration of GTP is higher inside the nucleus, and in this reaction Ran-GEF is catalyzes, or the Ran-GEF is catalyzes the reaction. But the number 2 pictures or the diagram shows that the import mechanisms, and in this case the importin is the important factor or important protein which helps in the import mechanisms of the components within the nucleus. And what is the function of the importin? Is important firstly the components or the protein or the signal peptides, whatever is that, binds with the important proteins, and then the importin transport inside the nucleus through the nuclear pore complex. Yes, this is the nuclear pore complex. Yes, and then the importin comes into the nucleus, and here the GTP is bound with the importin, and thus the signal peptides or the protein components, whatever needs to be imported inside the nucleus, is released, and then the importin is back to the outside of the nucleus, and here and obviously this process is occurred through the nuclear pore complexes, and here the Ran-GEF is helped in this process, and Ran-GEF converts GTP to GDP, and thus the concentration of GDP is higher inside the cytoplasm, and this phenomenon and this above phenomenon is maintained here. And then the number 3 diagrams or the nuclear export mechanism. Yes, this is the export mechanism, And in this case, the Ran-GEF firstly not worked. Firstly, the components which needs to be exported outside the nucleus bound with the exporting protein. Here the exportin is an important protein which helps to export the proteins or the components outside the nucleus. And first the GTP bound with the exporting protein complexes, and then the complex which need to be export is bound with this complex. And here this complex means the nuclear localization signal. The target protein is attached with the nuclear localization signal. The nuclear localization signal is a very important factor, because without the nuclear localization signal, any component cannot be imported or cannot be exported from inside to the outside of the nucleus, or from outside to inside of the nucleus. And then after forming these active complexes, this complex is exported with the help of the nuclear pore complexes we are in the outside of the nucleus. And then the complex is released from this. Then the complex means target protein with the nuclear localization signal is released from the GDP, as well as the GDP is released from the exporting protein, and then the exporting is come back to the nucleus. And thus the mechanism of the nuclear export is completed. But here is a different phenomenon, which is the mechanism of import of several transcription factor. And it is everybody known that the transcription factors are the very regulatory proteins, or very important proteins, which helps in the transcriptions, as because without transcription RNA cannot be produced, and the RNA without the RNA proteins cannot be produced. And that's transcription factors are the very important, and how they are import within the nucleus. The transcription factors like NF-kB and IKB, they are form a complex. And obviously here the nuclear localization signal, this is the nuclear localization signal, which is denoted by the green color, is bound with it. And then the phosphorylation is occurred. After phosphorylation, and phosphorylation is occurred with the help of the importin, and phosphorylation is occurred with the help of the importin. And then after the importin is bind with the this complex, then the complex is imported within the nucleus. But after bind the importin, this importin is proteolized by several proteasomal degradation pathway. But in this case, there is another different phenomenon is occurred. In this case, the yeast 4 protein, it is a transcription factor of yeast. Here the yeast transcription factor is bound with the nuclear localization signal, and then importin is bound with it, and dephosphorylation occurs, then the component imported within the nucleus. And this is the overall mechanism of nuclear transport. But now we started the mechanism, or we started to discuss the mechanism of transport of snRNA, means small nuclear RNA, and how the snRNA is transported from inside to outside of the cell, or from outside to inside of the cell, and how they formed complex with snRNP. And let's get detailed knowledge, and we'll start to discuss. Yes, this picture is clearly shown that the transport mechanism of small nuclear RNA means how the small nuclear RNA is transport from inside to the outside. And in this case, the small nuclear RNA firstly transported from the inside to the outside of the nucleus. And here the proteins which are important for binding or for forming a complex with small nuclear RNA is bound with the small nuclear RNA and forms the protein complex, which is known as the small and this protein is known as the small nuclear RNP. And after formation of the complex small nuclear RNP or small nuclear ribonucleoprotein, then these complexes come back or imported towards the nucleus, and thus the mechanism is completed. And today's topic is just about that. And I just end the topic of the nucleus today, and my next topic is a surprising one, and which is not related to the cellular organelles, and rather it is related to a several inventory or several surprising or several modern inventions of the about the molecular biology. Yes, welcome friends. Goodbye. Obviously, please subscribe my channel for more information by pressing the bell for getting the notification fast. Goodbye.